All right. Hey guys, good morning, good afternoon, wherever you are joining us from. Thank you guys so much for joining the Harvard Open Bio Final SRI Symposium. We're so excited to have all of you, and we've really been going over all of your research and all of the amazing achievements you guys have been, accomplishing over the past summer. And it's been really wonderful to see you guys grow as scientists, as students, as future change makers and leaders of the world. So we're so excited to be able to honor and recognize all of you and the work that you've been putting in these past few weeks. And I was just so excited to be able to start with our symposium. So my name is Zahra. I'm a rising sophomore at Harvard. I'm studying human developmental and regenerative biology. I'm the co-director for the lab team at Harvard Open Bio. So I'm mainly involved in, helping students run their own research projects throughout the year, giving grants, mentoring students on, running research or joining labs. So that's mainly what I've been involved in. And so, really excited to have you guys and, just reading over the research that you guys have been doing as well is really wonderful as, as part of the lab team. So we're so excited to really be able to honor you guys. So the way it's going to work, I'm going to be sharing a screen and I have all of your posters, all of your presentations. And so, one by one, I'll give you guys a time to speak and share your presentation, and then we'll keep going until our 5-minute break. And then from there, we'll come back and finish off. So let's get started. And sorry about my camera not being on. I have my computer, but it's not like my camera's not working, but I'm going to try to join on my phone after so I can have a camera as well. All right, do you guys all see my screen? Okay, awesome. So we're going to get started with our first presenter, Anusha. She's going to be presenting The Warrior Gene on Trial, a critical synthesis of MAOA, race, and environmental interaction.
For centuries, societies have asked the same question. Why do people commit crime? Some have searched for the answer in morality, others in social structures and biology. But the truth is, there's no single gene for crime that exists. Instead, the story is far more complex, woven from biological and environmental threads. Understanding these layers could transform how we prevent crime and how justice is served. Hello, everyone. I'm Anusha Singh. My mentor is Mike Bernari, and this is the Warrior Gene on Trial, a critical synthesis of the MAOE gene, race, and environmental interaction. From the devil to deprivation, our understanding of criminal behavior has changed dramatically over time. With genetic evidence on the rise, biology has become an even more urgent topic needing to be addressed. My research focuses on one of the most controversial of these biological factors, the low activity variant of the MAOA gene, coined as the warrior gene. This gene codes for an enzyme that breaks down key neurotransmitters such as serotonin, dopamine, and norepinephrine, which regulate mood, impulse control, and stress response. When the low activity variant of the MAOAL is present, these neurotransmitters are not broken down as efficiently. This changes the brain chemistry and can influence emotional processing, especially in the limbic system and the prefrontal cortex. Alone, this does not cause criminality, but paired with early life adversity such as childhood abuse or neglect, research shows a significant increase in the rise of aggressive or antisocial behavior. In my systematic review, I analyzed studies from um, PubMed, Google Scholars, and Galileo that examine not only the MAOA and maltreatment, but also how race, gender, and socioeconomic background influence its expression. I found consistent evidence for gene-environment interactions. The MAOAL can magnify the effects of trauma, but the supportive environment can buffer them. However, the picture becomes more complicated when you look at the demographics of the MAOAL frequencies, differing among the populations. For example, some studies found that 77% of the Maori males have the variant compared to about half of the Caucasian and Chinese males. These findings have been misrepresented in the media, leading to harmful stereotypes and even reviving echoes of eugenics. In the courtroom, the MAOA has been used in criminal defense to argue for reduced sentencing, the so-called warrior gene defense. This raises a huge ethical question. Does presenting genetic evidence in court shift responsibility away from the individual? Could it unintentionally reinforce racial bias in sentencing? My research suggests that without the proper safeguard, the misuse of science could do more harm than good. Ultimately, what I found is that MAOA gene is not a verdict of someone's future. It's a thread in a much larger web of biology, environments, and lived experience. The presence of the gene is neither destiny nor excuse. It's an opportunity to identify those risks early, to invest in supportive environments, and design a prevention strategy that are culturally sensitive and scientifically sound. If used responsibly, genetic knowledge like this could shift the narrative in our justice system from punishment to prevention. But if misused, it could deepen the very inequalities we hope to solve. The real challenge and responsibility now comes to us, the minds of the future, the minds that will power the future, and how we choose to apply it. Thank you.
Thank you so much, Anusha. That was a wonderful way to start us off. Really appreciate it. And now we'll get started with Jayan, who will be presenting his presentation on evaluating the use of microfluidic models for studying venous thrombosis through stress models.
All right, thank you. Can you hear me?
Yes.
All right. Hi, everybody. My name is Jane Tolia. My mentor was Sabrina Marnoto. Today I'll be walking you through my project. So, the number one cause of death in the world, as you may know, is due to heart and cardiovascular conditions. And the up to 50% of these conditions are caused by conditions such as thrombosis and atherosclerosis. The most important hemodynamic metric for measuring the progression of thrombosis and atherosclerosis is wall shear stress in these blood vessels because a low or too low or too high wall shear stress um, in these vessels can lead to the buildup of plaque or the formation of blood clots, which can be fatal. So it's important to get a sense of it's important to get a sense of how the wall shear stress in these models changes when responding to different stimuluses. And there's two ways to measure this. There's in vivo and in vitro studies. In vivo studies often involve taking animals or taking cadavers and measuring the flow of blood through there. And as you can imagine, that this is quite costly, ethically unsound, and it's very difficult to regulate. So experimentally, there are a lot of flaws in this. The second option for this involves the use of a technology called microfluidic chips. Essentially, these are chips that contain small milliliter or submilliliter diameter passages that can actually simulate the flow of fluid through them. So, for my project, I decided to use computational fluid dynamics software, CFD software, to simulate the flow of blood through one of these microfluidic chips to determine the viability of a chip for measuring thrombosis across age. And so what I did was I designed a model of a stenotic venule, a venule with a constriction in it in AutoCAD. Um, as you can see in figure one, this is a venule that expands and then constricts up to twice and half of its initial radius, which was its radius was initially zero point 0.1 milliliters or millimeters or 100 micrometers. It was then meshed with divisions of around 10 micrometers. And then I took a section of the channel and then used the CFD software OpenFoam to simulate the flow of blood through it. I simulated blood not as a Newtonian fluid, but as a Corot model. And this is significant because this is only a behavior that will greatly matter at the microfluidic scale with large amounts of shear stress. I ran 10 tests, each time scaling the model differently. So the radius of the model was initially 0.1 millimeters. I scaled it up 10 times to be 0.2, 0.3, 0.4, 0.5, up to 1 millimeter in radius, and then I simulated the flow of blood through it. And then I took the schematic of the wall shear stress and ran that through data post processing. And essentially, I did two things with that. We did two things with that. First, we identified the regions of highest and lowest stress. So we saw how the stress in these systems changed with the changing radius. And essentially, what we found was that the regions of highest and lowest stress were remarkably consistent. The region of highest stress was often in the most stenotic region, the region of the most change in wall radius. And the region of the least shear stress was often in the region of absolutely no wall radius changes, where flow was completely laminar and the no slip condition essentially was followed. The second thing we wanted to do was we wanted to see how the wall shear stress maximum and minimum actually changed with size. And this is where we found some interesting results. We found that not only did it consistently decrease across all metrics, we found that it often asymptotically approached zero and oftentimes followed an exponential decay pattern. And this is significant because modeling the flow of blood through venules across different sizes can help us understand how a person's risk for thrombosis and atherosclerosis changes across age. Because as a person's age increases, their blood vessels often dilate and increase in size and decrease in elasticity. So by modeling this factor and showing mathematically how the wall shear stress decreased and in exactly in what way it decayed, we can help researchers and medical professionals determine the true cause of a condition like thrombosis and atherosclerosis, which will in turn help us reduce heart disease and heart death um, in the future indefinitely. Thank you.
Thank you so much, Jayan. And now we're going to move on to our next presenter, Sayeda Rida, who will be presenting designing a multivalent peptide vaccine for Lassa lineages circulating in Nigeria using immunoinformatics, structural, and computational methods. Whenever you're ready.
Hello everyone, I'm Saida Rida Fatima and my mentor is Sir Onyeka from UCSD. I hope you're doing well. Now, basically, my research is about the design of a multivalent peptide vaccine for Lassa lineages circulating in Nigeria. Now, before moving on to the details, let's look into what exactly Lassa is. Basically, Lassa is a viral hemorrhagic fever that's endemic to West Africa and it's transmitted through contact with rodents or their excreta. Now, its symptoms can range from a mild fever to severe multi-organ failure. It has 7 lineages, and each of them are found in very different areas. So let's look into the first two figures of my poster. Lineage 1 is found in northeastern Nigeria. Lineage 2 circulates in southern Nigeria. Lineage 3 is found in north central Nigeria. Lineage 4 is often found in Sierra Leone, Guinea, and Liberia. Lineage 5 is often proposed as a distinct lineage, but could potentially be a sublineage for fourth. So it's found in Mali and Cote d'Ivoire. And lineage 6 is found in Mali and Cote d'Ivoire and Ghana. Lineage 7, which is the last lineage, is found in Togo, Benin, and Ghana. Now, moving on to why it is a significant public threat. Now, firstly, due to its high mortality rate, up to 15 to 20% in severe cases. Then there's misdiagnosis of asymptomatic cases. And then lastly, there's lack of effective vaccines or widely accessible treatments to this period. So, my approach towards this is actually making a multivalent vaccine. Now, how I did that? Firstly, I extracted the FASTA formats for all the 3 lineages from NCBI and Uniport. Then I used the IEDP epitope prediction tool to shortlist the B-cell epitopes through 2 different options. One, Bipipert lineage epitope prediction 2.0, and the second one, Kolaska and Tonga-Oka antigenicity test. Now, for shortlisting the T-cell epitopes, I shortlisted both MHC1, which is for CD4 helper cells, and MHC2 for CD8 cytotoxic cells. And then I shortlisted the best peptides, meaning the ones which had good peptide score and percentile rank, and checked for their antigenicity through Vaxogen 2.0 server, allergenicity through AlgPred and AllerCAC Pro, and toxicity through ToxinPred. After that, I organized my data and shortlisted the best epitopes and also made a list of the conserved epitopes and marked them on the glycoprotein structures using chimera X. Then I made the vaccine following the structure, which is mentioned in the screen. Adjuvant, linker, which was my flagyllene in this case, T cell epitopes, spacer, B cell epitopes, and histidine tag in the end for purification purposes. Now, following that, I predicted the vaccine structure on AlphaFold server and run a simulation on C and Sim to predict the immune response induced by these vaccines for 1 year with a prime and a booster dose in human body. Now, with that being said, these graphs right here show successful immune response of the body to the vaccine. The first graph shows how B cells went high and then were maintained. So basically, more B cells equals more antibody production. So better immunity. Second graph. LASV normally blocks interferons, which kind of makes the immune system blind to the antigen, but in the vaccine, that's not the case. As the antigen enters, the interferons increase, and then after the antigen is gone, they're wiped out around day 50. In the third graph, we have a sustained number of memory and non-memory Th cells, where memory Th cells are much larger in number because they're actually needed to activate the non-memory Th cells. Lastly, we have the graph that shows the spike in antibodies right after the dose, which was sustained later, which shows that there's durable protection and ongoing immune surveillance. So that's it from my side, everyone. Thank you all for your time and attention. If you have any questions, feel free to ask me in the Slack.
Thank you so much. Now we will have Aaron Choi, who's presenting Evaluating CAR T-cell Therapy for the Potential Treatment of Systematic Lupus and Erythromatopus. I hope I'm saying that right.
Okay. Hello, my name is Aaron Choi, and today I'll be presenting my research on evaluating CAR T therapy for the potential treatment of systemic lupus erythematosus. SLE, or lupus, is a severe and chronic autoimmune disease which is characterized by the breakdown of immune tolerance and the production of autoantibodies, where the patient's immune system mistakenly attacks its own organs and tissues, which causes a wide range of clinical symptoms such as kidney failure, blood vessel damage, and neurological symptoms such as seizures. Ultimately, the goal of SLE treatment is to achieve sustained remission, which is a temporary state of disease inactivity. However, current treatments like hydroxychloroquine, immunosuppressants, and monoclonal antibodies can reduce symptoms, but they don't selectively remove harmful or autoreactive immune cells, which also leads to broad immunosuppression. And that is the reason why remission rates currently remain low in SLE treatment. And because B cells play a central role in lupus pathophysiology, there has been an interest in targeted immunotherapies such as chimeric antigen receptor T cell therapy, or in the case of lupus, CD19 CAR T therapy. And this treatment extracts T cells from a patient, genetically modifies them via a viral vector such as a lentilvirus in order to express CAR receptors that target CD19, a B cell marker, and then re-injects them into the same patient to eliminate autoreactive B cells, which functionally resets the patient's immune system. In figure 2, the second generation of CAR T cells, which are the most commonly used, have specialized receptors which are made up of a single chain variable fragment, a peptide linker, a co-stimulator, which amplifies the signals received by the T cell, and an intracellular signaling domain, which activates the CAR T cell. And now for the review, my goal was to conduct a scoping review to evaluate the efficacy, safety, and remission rates of CD19 CAR T cell therapy in SLE patients. And we pulled data from recent case reports and pilot studies. Both the PubMed and American College of Rheumatology databases were utilized, and 5 search queries were run. And inclusion criteria were 1, case studies, which involved patients with refractory SLE, and 2, a follow-up of at least 3 months post-CAR T with results. And exclusion criteria were case reports with no remission results available and data from patients who discontinue CAR T therapy, and also non-English studies, duplicates, and reports that are related to non-human animals or diseases such as cancers that weren't related to lupus were also excluded. And overall, I identified 94 studies initially, and ultimately included 11 unique case or pilot studies, which covered 73 patients. And now the results were striking. We found that 71 out of 73 patients reached remission, which is a 97% success rate. In figure 4, the SLEDAI data, or disease status, of 17 patients showed that disease activity decreased and reached 0 in most patients within 3 months. And clinical remission was also paralleled by serological remission, as figure 5 shows that B cell counts decreased and then were replaced by healthy naive B cell populations, which shows the elimination of autoreactive B cell populations. And additionally, complement protein counts also decreased after infusion, which indicates that immune complexes were no longer being formed and deposited on organ systems and causing significant tissue damage. And also autoantibody counts such as anti-dsDNA also significantly decreased in the majority of patients. And additionally, in 2 studies, Yang et al. And Tan et al., they found that allogeneic CAR T induced remission in all patients, with none of the patients developing graft versus host disease, which indicates successful treatment outcomes and a potential for allogenic therapies to be implemented in the treatment of lupus. Uh, Moving on, another 2 studies, Yan et al. And Shed et al. Found that both, which measured both lupus-related autoantibodies and vaccine-induced antibodies before and after CAR T therapy. While lupus antibodies were eliminated, vaccine antibodies, like those for measles, mumps, and tetanus, were preserved, which suggests that CAR T therapy removes harmful CD19 B cells while sparing protective long-lived plasma cells that lack CD19, allowing patients to maintain immunity to prior vaccines. And overall, side effects were also generally mild. Most patients had only grade 1 cytokine release syndrome, with only one having grade 2 cytokine release syndrome with no other complications, mainly fever, which was the biggest symptom, which was resolved without intensive care. And a few of the patients had temporary neutropenia or mild infections, and none of the patients developed any severe neurotoxicity or other complications, which is huge because CAR T therapy is a very potent treatment, which has the potential for a lot of side effects. But the findings of the study proves that none of these patients really had any of those side effects, which solidifies like the potential treatment, like the potential effectiveness of CAR T therapy for SLE. And overall, these results are based on small case and pilot studies, which lack the statistical power of randomized trials and may be influenced by publication bias. However, we find that the consistency of the findings, such as rapid drug-free remission with minimal toxicity, is something that is just unprecedented in lupus treatment. And in conclusion, CD19 CAR T therapy shows very strong potential as a safe and effective potential option in the future for patients with severe treatment and treatment-resistant SLE. And future large-scale controlled studies and long-term follow-up will be critical to determine the long-term durability of remission and optimized treatment protocols in CAR T therapy. Thank you.
Thank you so much, Aaron. Now we have Samvid, who will be presenting translational feasibility of environmental biomaterials and bioabsorbents for in vivo PFAS sequestration, a critical review.
Nearly every person in this room has a class of indestructible man-made chemicals accumulating in their blood, with no way to get them out. My name is Samvid Govandani, and today I'm here to present my research on a potential solution. Okay, so per and polyfluoroalkyl substances, or PFAS, have earned the moniker of forever chemicals, and not without good reason. Their persistence stems from an exceptional carbon-fluorine bond with a dissociation energy of approximately 485 kilojoules per mole, rendering them highly resistant to natural degradation. The critical issue, as detailed in the top left of the poster, begins when these substances enter the human body. They become trapped in a cycle of enterohepatic recirculation, or EHC, leading to multi-year biological half-lives. This bioaccumulation is linked to a cascade of adverse health effects, ranging from endocrine disruption via PPAR agonism to hepatotoxicity, and where cellular damage is driven by oxidative stress through the activation of NF-kappa B and JNK signaling pathways. So, but despite all this, there's currently no approved clinical strategy for systemic PFAS detoxification. And this research gap establishes the central objective and the novelty of my work, which is to critically evaluate whether biomaterials that have been proven effective in environmental PFAS remediation can be used for safe in vivo sequestration in humans. To tackle this, I used the methodology detailed on the bottom left. A systemic review was conducted using the PRISMA framework, involving a comprehensive search across major databases like ACS Publications and PubMed. And after systematically filtering out the sources based on strict inclusion criteria, the process culminated in a thematic and critical synthesis of 138 peer-reviewed papers and articles. The results of the synthesis are visualized in the central figure on the poster, the feasibility efficacy matrix. This matrix categorizes materials based on their environmental absorption efficacy versus their biomedical feasibility. The matrix in Table 1 reveal a stark divergence. For example, materials like biochar and porous organic polymers are deemed not viable for human use because despite high environmental efficacy, their compositional heterogeneity and the potential for residual contaminants from pyrolysis, which is how they're formed, present unacceptable safety risks for in vivo applications. Conversely, if you look at the top right quadrant of the matrix, we can see our lead candidate, chitosan, because its molecular structure features positively charged amine groups that form strong electrostatic interactions with the anionic sulfonate or carboxylate head groups of the PFAS molecules, which is complemented by hydrophobic interactions. Cross-linking and enteric coating for improved absorption efficacy in the gastrointestinal environments is also very viable. But the most critical point here is that it's already established to be biocompatible, as is evidenced by existing FDA approval for medical uses. So that makes it a really feasible candidate for an oral binder to limit EHC. Following closely behind chitosan, we can see that alginate is a promising candidate for oral delivery due to its mild gelation properties and established safety profile for encapsulation. Meanwhile, silk fibroin's low immunogenicity and tunable structure and properties makes it a specialized candidate. Not as good as the first two, but ideal for developing stuff like extracorporeal filters. The key takeaway, therefore, is that the metrics for environmental success, like bulk adsorption capacity, are like poor predictors of biomedical viability. Instead, we need to look at specific properties like surface chemistry, degradation pathways, and interaction with biological components like plasma proteins, because these are the critical determinants for translating a material from environmental remediation to an in vivo clinical setting. This conclusion informs the roadmap for future research as detailed on the right of the poster. We must establish standardized in vitro protocols under simulated physiological conditions and construct toxicological assessments of the PFAS laden constructs. And this is because their biological interactions, the PFAS laden constructs, may differ significantly from the pristine biomaterials without the PFAS already absorbed onto their surface. However, the most crucial step is proceeding with the preclinical in vivo validation, and without that, this is the fundamental bottleneck in this field that's preventing the translational approach from going forward in clinical settings. Ultimately, this review's translational approach provides a novel framework to guide this new field, showing a clear path for transforming environmental science and to a direct tool for protecting human health. To conclude, I'd like to sincerely thank my mentor, Mr. Odera Ume, for his invaluable guidance and support throughout this project. Thank you.
Thank you so much. Now we will have Luca, who is presenting the mechanotransduction driven skeletal muscle hypertrophy and neural adaptations in track and field athletes, a systematic review.
Imagine a sprinter exploding off the starting blocks. In less than 10 seconds, they push their body to its limits, muscles generating immense force, tendons storing and releasing energy like cold springs, and neural circuits timing every contraction to the millisecond. This isn't just strength at work, it's powered by mechanotransduction, the microscopic process by which mechanical forces are converted into biochemical signals that drive structural and neural changes. The same mechanisms that make an athlete faster could help a patient recover strength after injury, slow age-related muscle loss, or restore mobility in neuromuscular disorders. My name is Luca Fabinger, and I'll present my findings from my systematic review on how mechanotransduction drives skeletal muscle hypertrophy and neural adaptations in sprint-trained athletes, and how these insights can be applied in training and rehabilitation. I followed the Prisma guidelines and searched on PubMed, Google Scholar, JSTOR, Europe PMC, and Semantic Scholar. As shown in Figure 1, I began with 106 studies. Exclusion criteria included non-human research, studies focused on cardiac muscle, long endurance, running, purely biochemical work without mechanical context, and others. After applying these criteria, 30 studies were selected for synthesis. First, Figure 2 illustrates three main mechanosensor models. The dragging model shows how membrane-bound lipids are pulled by deformation under high tension. This distorts channels like MSCL, opening a port and allowing ions to flow in. Next is the entropy model, and it works differently. Mechanical stress releases lipids bound to the channel surface, increasing protein conformational freedom and opening channels like K2P, which stabilize membrane potential during powerful contractions. The membrane dome model involves piezo proteins curving the cell membrane into a dome. Under tension, the dome flattens, mechanically opening the pore to allow cation entry. Though distinct in mechanics, all models converge on activating the MTR RC1, and IGF-1 pathways, both essential for protein muscle synthesis and hypertrophy. Sprint training with short, high-intensity bouts produces the sarcolemmal deformations that engages these sensors most effectively. High-tension loading enlarges type 2 fibers, increases pennation angles, stiffens tendons, and shifts fiber type towards type 2A. Neural adaptations include greater motor unit recruitment, higher firing rates, improved synchronization, and reduced antagonistic coactivation. One discussion point is the fiber type specific mechanosensor activation. Different fiber types may have uniquely sensitive thresholds, allowing targeted training to selectively enhance certain fibers, useful for both sprinting and rehabilitation. For rehabilitation, mechanotransduction principles could stimulate muscle regrowth and neural reactivation without overloading damaged tissue. Low-load, high-tension methods, such as blood flow restriction training, can activate mechanosensors while minimizing joint stress. In older adults, carefully calibrated resistance and plyometric work could improve tendon stiffness, counteract sarcopenia, and enhance balance through better neuromuscular coordination. A promising avenue is also integrated neuromusculoskeletal rehabilitation, combining mechanosensor-focused loading with neuromotor retraining. This dual approach restores not just muscle size and strength, but also timing and precision of force application. Advances in wearables and imaging could soon allow real-time monitoring of mechanosensor activation, enabling precise and adaptive exercise prescriptions. I would like to acknowledge the Harvard Open Biolab and my mentor, Sherelle Connor, for all the support that they've given me. And that's all for me. If you have any questions, feel free to ask me on Slack. Thank you.
Thank you so much, Luca. You did a great job. Now we have Raga, who is presenting the dystrophic heart, dilated cardiomyopathy as a cardinal manifestation of Duchenne muscular dystrophy and emerging treatments.
All right. Hi, everyone. My name is Raga Bukkaraju, and I was mentored by Aishwarya Iyer, PhD from U Maryland. And today I want to walk you through the cardiac complications of Duchenne muscular dystrophy, specifically how it progresses into a condition known as dilated cardiomyopathy, or DCM, and why our current treatments fall short. But more importantly, I'll highlight the exciting frontier of therapies that don't just manage symptoms, but aim to right the genetic basis of such a devastating disease. So what is DMD? DMD is caused by mutations in the dystrophin gene leading to the absence of functional dystrophin protein, which is why it's known as the most severe and common form of muscular dystrophy. While skeletal muscle degeneration is a hallmark, the heart is equally vulnerable and can manifest especially as early as the age of 6 years old. In cardiomyocytes, the dystrophin anchors the cytoskeleton to the extracellular matrix, providing flexibility and structure. And without it, mechanical stress during contraction in the heart causes microtears in the sarcolemma, triggering calcium influx, mitochondrial dysfunction and damage, and chronic inflammation, as seen in Figure 2 under current understanding. Over time, this cascade leads to progressive fibrosis, which is seen in 60% of mid-stage patients, left ventricular chamber dilation, and systolic dysfunction, clinically manifesting as dilated cardiomyopathy. This results in fatal conditions like heart failure, which is actually the leading cause of death in over half of DMD patients in their late teens. We can also note that calcium dysregulation has emerged as a central driver of DMD-DCM, with abnormal calcium handling, worsening cell apoptosis, and arrhythmogenesis. So let's take a look at the landscape of treatments tabs. Standard treatments like ACE inhibitors, beta blockers, and corticosteroids only offer partial protection. ACE inhibitors reduce afterload and fibrosis. Beta blockers blunt sympathetic overdrive, and corticosteroids delay skeletal decline. But none of these actually address the root cause, which is the absence of dystrophin and the downstream molecular chaos. The problem is that these treatments are reactive and not reparative. They don't halt disease progression, and their efficacy wanes in advanced stages. Moreover, they fail to account for the unique inflammatory and fibrotic signatures in DMD hearts, especially in female carriers and underrepresented populations, in which research is severely limited. If you take a look at Figure 2 under the Landscape of Treatments tabs, you'll see the comparison of new treatments to mainstream ones. The difference is that emerging treatments are restorative instead of symptom-based. Inflammatory reprogrammers such as NFKB inhibitors or IL-6 blockers target the chronic immune activation that accelerates cardiac decline. And with calcium dysregulation now recognized as a key culprit, ion channel modulators such as ryanodine receptor stabilizers are being explored to restore calcium homeostasis and prevent arrhythmias. Mitochondrial transplantation allows us to implant viable mitochondria to restore the bioenergetics of the cell and restore contractile forces. Cell-based approaches like cardiosphere-derived cells and mesenchymal stem cells operate via paracrine signaling that reduces fibrosis and actually promotes regeneration of cells. And what promises to be the most potent therapy of all is gene editing. Therapies such as CRISPR-Cas9, which you guys have probably heard about, and exon skipping actually excise hotspot exons of the DMD gene, such as exons 45 to 55, and aim to restore fully functional dystrophin expression. If you take a look at the conclusion tab, you'll see that not only do emerging treatments have much better effectivity, but that calcium dysregulation has also been found as a main driver of DMD-DCM, as explained before. This is why our review has found that emphasis has been placed on making ion and calcium channels main therapeutic entry points. However, limitations like standardization of care across patient populations, including female and late-stage patients, combinatory therapy protocols, and longitudinal monitoring of patient health post-treatment, considering multiple factors like exercise, diet, etc., still remain. And these gaps are what constitute the future of DMD-DCM healthcare, bringing us one step closer to accessible treatment for all. Lastly, I'd like to thank my mentor, Aishwarya Iyer, for their unwavering guidance and insight throughout this 8-week program, and to the OpenBio staff to let me explore, grow, and contribute to meaningful research. Thank you all for your time, and if you have any questions, I'd be happy to answer them in the Slack. Thank you.
Thank you so much, Raga. And now we have Gautam.
Is this slide gonna? Okay, there we go. Thank you. Hello, everyone. My name is Gautam Sathi, and today I'm excited to share a review on antimicrobial polymer coatings, or AMPCs, which I wrote under the mentorship of Anand Dikshit. I'll be honest, when I first started this manuscript, I thought I was just going to be researching cool ways to kill germs. But it turns out the real challenge isn't finding new germ killers, it's figuring out how to compare them. Imagine you're at the grocery store and there's 50 different kinds of apples, but none of them have labels saying if they're sweet, tart, or good for baking. That's pretty much the situation with AMPCs right now. So what exactly are AMPCs? They're polymer coatings that have been combined with antimicrobial agents to stop bacteria and other microbes from growing on their surfaces. They're becoming a really big deal in a lot of industries, including healthcare, food packaging, textiles, and infrastructure. In healthcare especially, they can be used on things like surgical tools and catheters to reduce hospital-acquired infections, which affect about 100 million people globally each year. AMPCs are also used in food packaging to prevent spoilage and extend shelf life, and in textiles like personal protective equipment to improve long-lasting hygiene. AMPCs work in a few different ways, depending on the type of agent used. Some agents, like silver nanoparticles, release ions that kill microbes. Others, like quaternary ammonium compounds, or QACs for short, work through contact killing. And some, called zwitterionic polymers, prevent microbes from even sticking to the surface in the first place. The problem is that with so many different types of agents and mechanisms, it's really hard to compare their performance. The scientific community uses a bunch of different testing methods, each with their own strengths and weaknesses, making a cross-study comparison extremely challenging. This makes it feel a little bit like comparing apples to oranges, or, comparing how fast a car drives to how well it can flow. That's where my review comes in. To tackle this issue, we performed a structured literature review analyzing hundreds of peer-reviewed studies to identify the most used agents and testing methods. We focused on 8 of the most commonly used antimicrobial agents, 5 major testing methods, which we identified based on their frequency of use in the literature. We then organized the data by agent and testing method to create a comparative framework. This allowed us to find general trends and figure out which agent works best with which testing method. Our findings showed some interesting trends. In general, agents like silver nanoparticles, or AGNPs, and QACs performed well across the board in most standardized tests, with AGNP showing high performance across all methods. However, AGNP and other ion-releasing agents like copper and zinc oxides can be concerning because of their potential cytotoxicity and environmental impacts. On the other hand, Zwitter polymers and other anti-adhesion agents were weak in kill-based tests but excelled in long-term biofilm resistance, which is much real world scenarios. Meanwhile, agents like polyepsilene offered a great middle ground, showing strong performance similar to AGNPs but with better biocompatibility. Ultimately, this review highlights a critical gap in research and the desperate need for standardized testing frameworks that can fairly compare all these different types of agents. Our paper serves as an early step toward filling the gap by providing a foundation for future research and development of AMPCs. As these coatings become more and more common, having a reliable and consistent testing method will be essential to ensure public health and safety. I'd like to end off by thanking my mentor, Anand Dikshit, for his constant support, guidance, and detailed feedback throughout the process of writing this paper. His insight was invaluable at every stage. I'm also extremely grateful to the Harvard Open Bio staff for organizing this incredible program and dedicating their time over the summer to create such a meaningful experience for us. A special thanks to Sahil Sud and guest speakers for delivering engaging lectures and introducing us to exciting new topics. That has truly been truly been an unforgettable experience, and I'm extremely thankful for being a part of it.
Thank you so much, Gautam. Wonderful job. Now we have Vera.
Hey, so good morning, everyone. My name is Vera. My mentor is Olivia Carmen, and today I'll be presenting my narrative review, which is the behavioral manifestations and neurophysiology of emotional dysregulation in kids with ADHD and ASD. So, what is ED and why does it matter? Emotional dysregulation, or ED, is the inability to effectively regulate or react to emotions, and it's an especially common feature among children and adolescents with psychiatric disorders. This can involve lack of awareness or acceptance of emotions, inability to regulate the speed or intensity of escalation, or struggling to return to an emotional baseline. ED often manifests in maladaptive behaviors like mood instability, temper tantrums, aggression, irritability, or excessive reactions. These behaviors kind of reflect a limited set of problematic strategies that people use to relieve intense emotions. But these strategies actually worsen their experience and creates a vicious cycle that fuels further maladaptive behavior. Now, ED is not a disorder in and of itself, but it's highly prevalent across multiple conditions, and it's present in up to 45% of pediatric psychiatric patients. If left untreated, it increases risk for self-harm, substance use, suicide, and chronic health problems later on. And kids with ADHD and ASD have especially high rates of ED, which is why we decided to compare ED and both to inform future interventions. So ADHD and ED. ADHD is defined by impulsivity, hyperactivity, and or inattention according to the DSM-5. However, earlier editions listed ED as a core symptom, but later versions kind of shifted focus to executive function and inattention, which caused ED to be overlooked in both assessment and treatment despite increasing evidence that kids with ADHD often have ED. Behaviorally, ADHD-related ED often appears as rapid, intense, externalized reactions like temper outbursts, verbal aggression, tearfulness, or irritability. These are often mistaken for misbehavior, which can lead to outside consequences like rejection, disruption, or disciplinary action. And it kind of fuels the ADHD maladaptive cycle, as we see in figure 1 over there, as these lead to negative emotions like frustration or anger and results in more maladaptive manifestations of ED. Neurophysiologically, researchers have also found that dysfunction in the prefrontal cortex, amygdala, and anterior cingulate cortex impairs the ambidaural control and heightens reactivity. Both the bottom-up and top-down processes are affected, and reduced white matter activity has also been found, which is something that weakens integration between emotional and cognitive regulation. So what about ASD? ASD involves social, sensory, and communication deficits plus restricted repetitive behaviors. Behaviorally, ED and ASD includes irritability, meltdown, self-injurious behavior, and difficulty recovering from distress. Triggers can include sensory sensitivities, communication challenges, and social misunderstandings. Like in ADHD, research suggests that these environmental challenges lead to externalized responses of ED, which can cause social misunderstandings, rejection, and limited opportunities for social learning, thus exacerbating the challenges that children with ADHD face and contributes to further ED, like we see in figure 2. In addition to externalization, some people with ASD also have a high rate of internalizing, which exacerbates social impairment. Emotional suppression can lead to internalized stress, which in turn contributes to emotional burnout and social burnout and further internalized ED. Neurophysiologically, ED and ASD also involves atypical bottom-up and top-down processing, with reduced white matter integrity and disrupted amygdala prefrontal connectivity. This impairs integration, again, between emotional and regulation systems, especially during fear processing. So how is ED and ADHD and ASD similar, and how is it different? Well, both ADHD and ASD have ED they involve the same neurophysiological dysfunction in the same areas that control social emotional functioning and regulation, as well as similar outside consequences, but they differ in the expression. ADHD is primarily externalized, while ASD is a mix of externalized and internalized patterns. This creates different maladaptive cycles for each, as you can see in my figures. In ADHD, ED presents as outward reactivity, which often results in outside consequences, leading to more intensified negative emotions and subsequent dysregulation. In ASD, ED typically manifests as externalized or internalized responses, which exacerbates different stressors or outside challenges that leads to ED in the first place, forming a maladaptive cycle. And why is this research important? Well, this research is important because ED is a pervasive and under-recognized feature of both ADHD and ASD, and it drives cycles that can harm social, academic, and emotional well-being and functioning. Future research should develop interventions targeting both behavioral and neurophysiological aspects, adapt strategies for schools, and consider reinstating ED as a core diagnostic feature for both diseases like it was. While behavioral and pharmacological treatments have been developed, specifying treatment through the different lens of the expression and the maladaptive cycles of each disorder would be useful, because there haven't really been made any targeted interventions towards any specific disorder, only towards ED as a whole, or they've also used drugs intended for ADHD or ASD to try to lessen the effects of ED. Recognizing ED early will be able to enable earlier treatment, reduce impairment, and improve long-term adaptability and well-being in affected youth. Thank you for listening, and please let me know on Slack if you have any questions.
Thank you so much, Vera. Wonderful job. Now we have Rishi Ranganathan, who unfortunately could not be with us today. So instead, I'll be playing a recording that he recorded for all of you. So just give me a moment to pull that up. Can you all see my screen with the video? All right, awesome.
Hello, my name is Rishi Ranganathan, and today I'll be presenting the findings of my literature review, Income Inequality and Access to Primary Care, a cross-national review of structure.
Sorry, you guys can't hear the audio, or is it frozen?
We could hear it in the start, but we can't hear it anymore.
Yeah, it's like totally frozen.
Okay, let me see what I can do.
Care reduces. Hello, my name is Rishi Ranganathan, and
Can you
today
guys hear it now?
the findings of my literature review, Income Inequality and Access to Primary Care, a cross-national review of structural barriers in high-income countries. The central
Yeah.
question I explored was this: how does income inequality?
Wait, could you guys hear
Is
that?
there like for a lot of people, I can hear it, but the screen's kind of like still frozen. I can hear it.
Okay, let me put the subtitles. I guess there's no subtitles to it, but it's a little blurry itself, the video. So as long as you see him talking like in the top right screen, it should be fine. But you guys can hear the audio well?
Yeah.
Okay. All right, just message me in the chat if
Rishi
anything.
Ranganathan, and today
Thank you so much, guys.
Income Inequality and Access to Primary Care, a cross-national review of structural barriers in high-income countries. The central question I explored was this: how does income inequality influence access to primary care and preventative services in undeserved populations within high-income countries? While many of us assume that universal healthcare coverage ensures equal access, my research found that this isn't necessarily true. Even in countries with public healthcare systems like Canada, the UK, or Germany, low-income populations continue to face significant and persistent barriers when it comes to accessing primary care. Primary care is not just about routine checkups. It's the front line of disease prevention, chronic illness management, and
Hey guys, so I'm going to try to figure out the audio, but in the meantime, you guys can take the 5-minute break a little bit early. So we'll be back at 1:27 and then we can play the video. Hey guys, I think I fixed the issue with the audio, so I can just I'm gonna let the video continue playing, and then right after we could start with the break. And thank you so much, by the way, for the clarification. So we'll resume at 1:35, like with everybody else.
care access and health disparities. I also used backward citation tracking from a major systematic review published on socioeconomic inequalities in physician visits. In total, I included over 40 high-quality empirical studies and systematic reviews published between 2010 and 2024. My inclusion criteria were clear. The studies had to examine access to primary care or preventative care, compare groups by income or SES, and focus on general or undeserved populations rather than disease-specific ones. Across the literature, three major findings emerged. First, there is a strong and consistent evidence that income-based disparities in access to primary care exist in nearly all high-income countries, even those with publicly funded healthcare. For example, studies in Canada, Germany, and the UK showed that lower-income groups use primary care services less frequently, face longer wait times, and often delay or avoid care altogether, even when it's free at the point of use. This tells us that financial access is only one part of the equation. Structural barriers such as limited appointment availability, provider shortages on lower income areas, and lack of transportation also play a massive role. Second, the review highlights that income inequality intersects with other forms of marginalization, including race, gender, immigration status, and geography. For example, low income immigrant women may face for example, low income immigrant women may face cultural mismatches with providers, language barriers, or institutional discrimination. These overlapping disadvantages create unique access barriers that go beyond just income. Unfortunately, most studies still treat income as an isolated variable and fail to account for these intersectional dynamics. This limits the development of effective and targeted interventions. Third, many studies focus heavily on insurance coverage or other number of doctor visits, but neglect measures of quality, continuity, and patient experience. That means we don't always know if increased access actually results in better health outcomes, especially for undeserved groups. Also, several gaps became apparent during this review. First, lack of longitudinal studies. We don't have enough data on how access changes over time or during economic shifts. Two, limited qualitative research. Most studies rely on quantitative data like surveys and claim records, but we're missing the live experiences of those facing barriers. Three, insufficient evaluation of policy outcomes. While policies like Medicaid expansion have shown promise, we lack evidence on their long-term effects and scalability. So what does this mean for policy? While expanding insurance coverage is essential, it's not enough. The literature makes clear that we need multidimensional reforms, including better distribution of providers in undeserved areas, appointment systems that accommodate working class schedules, cultural and linguistic training for healthcare providers, and community engagement and intervention design. Some promising models like patient-centered medical homes and community health workers have shown potential when combined with outreach and support services. But without structural reforms, even well-meaning policies will fall short. In summary, this literature review underscores that income inequality continues to be a powerful predictor of unequal access to primary care. Even in the world's richest nations, financial barriers are important, but they exist alongside deeper structural and cultural barriers that must be addressed simultaneously. Addressing these disparities isn't just about equity, it's about sustainability. Without access to preventative care, low-income communities will suffer worse health, greater disease burden, and higher costs. It's both a social justice issue and a public health imperative. To build fair and effective
All right, guys, this marks the end of our first round, so we can take our break and we'll resume at 1:35. All right, guys. Next, we have Pavan, who also could not be here today, so we'll be playing a recording of his presentation.
Hi, my name is Advit, and I'll be presenting my research on.
All right, it seems like Pavan is actually here. It's Admit who's not here, so we're going to play his video.
Hi, my name is Advit, and I'll be presenting my research on the effects of climate change on vegetation in marsh wetland ecosystems. I chose this topic because marsh wetlands are one of the most ecologically important ecosystems on this planet. They support biodiversity, improve water quality, and help protect coastlines. And a lot of this depends on their plant life. But as climate change continues to intensify, the vegetation in these systems is becoming more vulnerable, and the ecosystem services marshes provide may be at risk. Marsh vegetation plays a vital role in ecosystem function. It regulates water flow, stores carbon, and provides water and shelter for both aquatic and terrestrial organisms. However, as climate change accelerates, it's starting to impact both the productivity and structure of vegetation in marsh wetlands. Temperature increases, changing rainfall patterns, and rising sea levels are altering plant growth, carbon storage capacity, and even the species composition in these ecosystems. These shifts could have long-term impacts on wetland stability and function, making it crucial to understand the relationship between climate stressors and vegetation response. To explore this topic, I conducted a literature review using the Wiley Online Library database. I began with a broad search that returned over 11,000 words. Articles. From there, I applied several filters to narrow down the pool to the most relevant studies. I excluded anything published before 2015, articles that weren't peer-reviewed or in English, and studies outside the field of environmental science. After this filtering process, I was left with a total of 359 articles. Then I selected those that focused specifically on the interaction between marsh wetlands, vegetation, and climate change, and were most relevant to my topic. From these groups of studies, I identified 4 key themes that describe how marsh vegetation is being affected by climate change. Temperature-driven shifts. Warming temperatures have altered marsh vegetation growing patterns. In some cases, higher temperatures have increased productivity by lengthening the growing season. However, this has also led to earlier dieback, reduced carbon sequestration efficiency, and changes in gas exchange, such as higher methane release, which could worsen greenhouse gas concentrations. Hydrological stressors. Precipitation changes in sea level rise affect marsh plant communities by altering flooding patterns, salinity, and sediment delivery. These changes can reduce species diversity and shift the competitive balance between native and invasive species. Climate change is also affecting the types of plants that dominate these areas. Some native species are being replaced by more heat or salt-tolerant plants. This shift can change how the wetland functions. For example, some species may not be able to stabilize the soil or filter the water as effectively as others. Last but not least, regional variation and resilience. One of the most important findings was that response of marsh vegetation is highly variable. In some areas, plants show strong resilience to climate stress, while in others, even small changes in sea level or temperature lead to major declines. This suggests that the vulnerability of marshes depends on both environmental conditions and human impacts, like land development or pollution. In summary, climate change is affecting marsh wetland vegetation in both subtle and dramatic ways. These changes influence how carbon marshes can store, how well they protect coastlines, and how they support biodiversity. These impacts aren't the same everywhere, which means that climate adaptation strategies need to be region-specific and based on detailed local monitoring. Moving forward, we need more long-term, site-specific studies to understand how marsh vegetation will respond over time and what we can do to protect or restore these ecosystems. I'd like to thank my mentor, Shan Dixon, for her guidance and support throughout this project, as well as the Harvard Student Research Institute for this opportunity to conduct this research. Thank you.
All right, awesome. So now we're going to move on to our next presenter.
My name is
We have Maggie who will be presenting next.
Multiple myeloma, MM, is a cancer of plasma cells, a population of blood cells best known for generating antibodies. Plasma cells are terminally differentiated B lymphocytes and reside within the bone marrow. MM disease pathology is marked by the evasion of cell death, proliferation of malignant plasma cells, and subsequent increased levels of monoclonal antibodies. The progression of MM is compartmentalized within 3 stages. Please direct your attention to Figure 1. The first, monoclonal gammopathy of undetermined significance, or MGUS, involves heightened serum and urine immunoglobulin levels within patients, while lacking any evidence of malignancy. Next, in smoldering multiple myeloma, SMM, the patient embodies a precancerous condition where an abnormal plasma cells proliferate at an increased rate within the bone marrow and contribute to the overall increase of M protein. Lastly, in fully fledged MM, the disease is demarcated by the CRAB criteria: hypercalcemia, renal failure, anemia, and osteolytic bone lesions. Please direct your attention to Figure 2. BCLXL, BCL2, BCLW, MCL1, and BFL1 are anti-apoptotic proteins that prevent effector proteins back from inducing apoptosis. However, pro-apoptotic sensitizer proteins NOXA, BMF, HRK, and BAD neutralize the aforementioned BCL2 proteins and release the pro-apoptotic activator proteins PUMA, TBID, and BIM. These activator proteins activate BAX and BAC on the mitochondrial membrane, leading to caspase activation and ultimately cell death. This is illustrated in Figure 3. The regulation of apoptosis is entirely dependent on the threshold between pro-apoptotic and anti-apoptotic proteins in vitro. Many MM cells overexpress anti-apoptotic proteins to avoid cell death. MM also obtains proliferative capacities. During B cell development, aberrant translocations can occur, where cyclin D is moved from its original chromosomal locus to the IGH1 locus, hence allowing it to be continually expressed. Cyclin D allows myeloma cells to replicate and divide on an uncontrollable and unprecedented rate. Approximately 35,000 people are diagnosed with MM each year. As it stands, there is one FDA-approved regimen that all MM patients are eligible for, and that's treatment LEN. Most patients are eligible for auto stem cell transplant in an attempt to repopulate the patient's bone marrow with new immune cell populations. Auto SCT is combined with maintenance therapy, where patients are monitored for the reemergence of malignant plasma cells. Negativity status might be maintained for years, as some patients can be stratified into low and high risk for disease progression. For example, combination therapies such as RVD, Revlimid, also known as lenalidomide, bortezomib, and dexamethasone, are available to high-risk patients. Additional lines of therapeutics include venetoclax, navitoclax, ABT-737, and novel proteasome inhibitors. Many of these treatments are set out to induce apoptosis in cells that overexpress anti-apoptotics. While I have described many advancements in the treatment of MM, unfortunately many patients will either become resistant to therapy or progress to late-stage myeloma. A keynote treatment that is a current area of intense research is CAR T-cell therapy. Please direct your attention to Figure 4. During CAR T therapy, millions of the patient's T cells are extracted via blood draw. CAR T plasmids are designed to target antigens that are uniquely expressed on a cancer cell, along with T cell receptor components, to generate a T cell receptor that can target these aberrant cancer cells for death. These T cells are further grown, and the cells are infused into the patient's bloodstream. While this technology has seen recent promise, there are several caveats to this treatment. Antigen escape can occur when a malignant cell fluctuates its surface antigens in order to bypass the CARs of T cells. On-tumor off-target effects occur when therapeutics target proteins that are present within tissues outside of the cancer target. Most importantly, patients can experience CRS or cytokine release syndrome that can produce many unwanted immune side effects or even result in death. Moreover, one round of CAR T-cell therapy costs roughly $500,000, hence deeming such therapies as financially unsustainable. In the future, I aspire to inform patients about the side effects, reduce the cost implications, and increase the accessibility of treatment. I would like to acknowledge my mentor, Veronica Narte, for all the support she has given to me during this project. Thank you.
Thank you so much, Maggie. That was a wonderful job. Now we have Sophie who will be presenting her project.
So my project, our goal was to simulate cataract vision in rainbow trout, specifically looking at how it might affect their foraging and predator evasion abilities. And so we found that most studies on cataracts and fish were about farmed fish, so we wanted to look at wild fish and potentially create a model that we can use for other species to evaluate their if they if other species of fish got cataract, how we can evaluate that. So, what is visual acuity? So, visual acuity is how well the eye can distinguish fine details and spatial patterns in the environment. And one way we measure that is using cycles per degree, or CPD, where one cycle is a pair of black and white bars per 1 degree. And so the higher CPD you have means that you can see more details. So in humans, 20/20 vision is about 60 CPD, and in rainbow trout, that is 4.29 CPT. So they have much lower visual acuity compared to humans. And cataracts are a condition where proteins clump in the lens, and that causes scattering of light. So light isn't able to reach the retina in a normal way because it's scattered, and that also causes a clouding of the lens. And so how we simulate that is there's a program called Acuity View in R. So it takes the CPD and distance of an image, and it uses a Fourier transformation to simulate what a trout might see. So using the trout's CPD, we used a prey image of a stonefly and a predator image of an otter, and we modeled different distances from 0 feet, 1.2 feet, 3 feet, and 5 feet to simulate what a trout might see. So that's the figure on top. And then to simulate cataract vision, we used GIMP and we used a gray overlay with different percentage opacity to represent different severities of cataracts based on a grading scale used in a prior paper. So what we found is that at 1.5 feet distance, the stonefly begins to lose fine details, and at further distances, so 3 and 5 feet, it becomes almost indistinguishable from the rock that it is sitting on. But overall, the otter remained the same, which suggests that larger predators are easier for rainbow trout to detect. And then for cataract, we found that at 75% severity, it's pretty difficult to make out details, suggesting that severe cataracts can significantly reduce a trout's ability to hunt and detect predators, which can lead to starvation and being hunted. But there are limitations of this research, as Acuity View can't simulate motion detection or even colors, as rainbow trout have been known to be able to see colors in the UV spectrum that humans can't see, so we can't simulate that. And also might not fully represent what cataracts vision is, as cataracts can be localized compared to affecting the entire lens, which is what we simulated in this study. But despite these limitations, this study provides a valuable first step towards understanding how cataracts may affect wild fish populations and give us insight into ecological consequences and help us and provide information to create conservation in the future. Thank you.
Thank you so much. Now we have Torun who will be presenting.
All right, so good morning. My name is True Malarvison. My project is on bistable auxetic stents. So first of all, in blocked arteries, stents are commonly used. Essentially, conventional stents are used within arteries to expand the overall arterial radius, and this essentially improves overall arterial flow. The issue with conventional stents is that they have a 60% chance of restenosis, or the re-narrowing of an artery after the stent placement. This can cause multiple issues such as limited oxygen flow to vital organs, as well as the need for overall stent placements to be placed over and over again. Essentially, what this project aims to do is utilize auxetics, specifically bistable auxetics, to treat this using customized vascular conformability techniques. then using machine learning models to improve the overall patient customization of the stent, and then finally optimize the overall stent performance utilizing strength simulations. So first of all, what are auxetics? So auxetics are classified as materials or microstructures that can expand in both its vertical height as well as horizontal length when stretched. You can see pictured in the top section in the blue images, we have an example of a contracted versus expanded state of an auxetic material. Auxetics also have a bistable nature, meaning they have a contracted and expanded state, and they actually be tweaked using two variables called the t and theta value, as pictured in the top right. You can see a low t and a low theta values expansion rate versus a high t and a high theta value expansion rate. So if you move on next to the methodology section, we actually need to know where in the patient the stent needs to be placed so that we can customize each stent to the patient. This leads us to our coronary artery segmentation model. Essentially, what this does is it takes a 2D XVA scan and can segment the various areas of the scan. So here we can see versus the input and output image. In the green, we have a highlighted healthy region. In the red, we have a stenotic region, and in the blue, we have a thrombotic region. So now that we know where in the patient in a 2D volume that the stent needs to be placed, we now need to approximate that into 3D volume so that we can actually make the overall auxetic stent. In the next section of methodology, under our input data section, we can see that we have our secondary machine learning model. This is trained on a KUKA data set. This utilizes 3D CT scans. And since CT scans are 2D layers or 2D images in multiple layers, we can essentially parse through each layer and identify a cross section or segment a cross section and add it all up using a visualization toolkit called Kitware, and we can approximate overall 3D volume. The next step is actually making the auxetic unit wrap around overall volume. That can happen in the image you can see in the third section of methodology. And finally, actually generating the overall model and simulating it in FEA testing, which is finite element analysis. Here we're essentially using computational fluid dynamics, and we're simulating a turbulent blood flow, and we're essentially seeing how the stent performs up against a turbulent blood flow. All the way towards the right, you can see what an actual stent would look like under the scenario. You can see that the stent is also perfectly customized to an example patient. You can see it forms all the ridges and values. Next is the results and validation section. Our segmentation, or the coronary artery segmentation model, had an overall accuracy of 83%, which is very good for a 3,000 image data set, as well as an F1 score of 0.83. This is all done through Matplotlib, as well as our second essentially result and validation test. This is auxetic design results. Here we're testing the 6 various auxetic designs explored throughout this project just to see the overall performance of each design. Here we can see that using a simulated testing stand and testing for von Mises stresses, we can see that the trichiral design, or the 5th pattern in the table listed below, performed the best and had the least amount of von Mises stresses. Next is our FEA validation. In the first graph at the top, we have radial principal stress distribution graphed. Here we can see that auxetic stents have a lower peak stress distribution, as well as a 32% lower peak stress overall, which can potentially show its improvement against conventional stents, as well as a 50 million life cycle, a fatigue life comparison versus a 10 million life cycles of conventional stents. Finally is our blood flow comparison. We have a normal versus auxetic stent all the way to the bottom right. Here we can see that auxetic stents have a lower average turbulence, which indicates it's a higher blood flow rate, as well as less variation along its curve. So overall, the conclusion of this project was that the auxetic stent proved to have a more uniform force distribution when compared to conventional stents, and can last for a longer 5 times longer life cycle strength, as well as a lower peak stress of 32%.
Thank you so much, Tarun. Now we have Zoha who will be presenting.
The most dangerous cell in rheumatoid arthritis isn't an invader. It's one that was born to protect the joint but becomes reprogrammed into a persistent driver of inflammation and tissue destruction. Rheumatoid arthritis, or RA, is not simply a more inflamed version of osteoarthritis, or OA. It is a chronic autoimmune condition where the immune system misdirects its attacks toward the joints, triggering persistent inflammation and irreversible tissue damage. At the heart of this process are fibrillars like synoviocytes, or FLS. In healthy joints, FLS maintain structural integrity, secrete protective factors, and regulate inflammation. In RA, however, they undergo a massive transformation. They become aggressive, inflammatory, and resistant to apoptosis. actively degrading cartilage and orchestrating immune cell recruitment. But what causes this transformation? A major factor is epigenetic reprogramming, changes in DNA accessibility, and gene expression patterns that alter cell behavior. However, the specific transcription factors that drive this reprogramming in REFLS are not well understood. So, our objective was to identify disease-specific transcriptional regulators by integrating RNA sequence and ATAC sequence, a multi-omic approach, to reveal how FLS from RA patients differ from those in OA controls. The differences between RA and OA FLS were robust. Principal component analysis of gene expression data showed complete separation of the two diseases as seen at the top left corner of the results. REFLS displayed strong upregulation of inflammatory mediators, including IL-6, IL-1B, and TNF, as well as Toll-like receptors. OAFLS expressed genes linked to extracellular matrix maintenance, oxidative stress protection, and metabolic regulation, consistent with a reparative rather than inflammatory role. Chromatin accessibility analysis told a similar story. In RAFLS, immune-related enhancers and promoters were more accessible, indicating these cells are primed to sustain inflammation. OAFLS lacked this epigenetic readiness, aligning with their more homeostatic function. When we further ranked these transcription factors, three stood out in RA. STAT1, which is part of the NFKB1 family, and IRF1, central players in immune activation. In OA, the most dominant regulators were CEBPA and PPARG, which coordinate tissue repair and metabolic homeostasis. Network analysis revealed that RA regulatory architecture was densely interconnected, as seen at the top right corner of the results section, under Figure 2, with multiple feedback loops ensuring inflammatory signals persist even without continuously external triggers or stimuli. OA networks were fragmented, reflecting lower regulatory redundancy and a focus on maintenance rather than escalation. To generate these insights, we used the TIG Systems biology platform, which integrates RNA sequence, ADEX sequence, and 3D chromatin interaction data to build a greater genome-wide regulatory network, as seen at the bottom left of the poster under TIG pipeline. It maps transcription factor binding motifs to principal chromatin, links them to their target genes, and uses a PageRank centrality algorithm adapted from Google search ranking to identify the most influential regulators in the network. In brief, RA fibroblasts are stably reprogrammed by master transcription factors such as STAT1 and RELA, amplifying inflammation while dismantling homeostatic programs, whereas OA fibroblasts retain reparative networks, as highlighted under conclusion on the right side of the poster. Mapping these divergent regulatory architectures further not only highlights therapeutic targets by re-educating RA cells, but also provides a framework for next-generation spatial and longitudinal profiling. Thank you.
Thank you so much, Soha. Now we have Rishi.
Hi, my name is Rishi, and today I'll be discussing the evaluation of prompt engineering techniques on large language model explanation quality in clinical question answering. It's estimated that every 73 days, medical knowledge doubles, making it incredibly difficult for clinicians to keep up with the influx of information. To combat this, LLMs, or large language models, with their ability to process and synthesize vast amounts of medical data, have recently emerged as a promising solution for supporting physicians in healthcare settings. However, their implementation carries serious concerns about the quality and transparency of their responses, especially for medical Q&A tasks. Existing research in this field focuses on evaluating primarily answer accuracy, but in clinical settings, this isn't enough, as the why behind an answer is just as, if not more important. Faulty justifications, even if followed by right answers, can increase riskiness and reduce physician trust in responses. In a field as high stakes as healthcare, having this interpretability is critical. To address this gap and optimize clinical utilization, I explored how different prompt engineering frameworks influence the explanation quality alongside accuracy of LLMs on the MedXQA benchmark. In Figure 1, we can see the overall pipeline for this experiment. I utilized MedXQA, a novel medical Q&A benchmark with examples from underrepresented medical specialties. I then tested several prompt engineering strategies on these dataset questions and asked LLMs to generate explanations for their answers. These prompting strategies involve refining instructions given to LLMs to improve the effectiveness, relevance, and reasoning quality of model outputs without changing the underlying model architecture or training data. For example, in this study, I tested few-shot prompting, where I included reference examples from MedXQA to guide explanations. Agentic frameworks take this a step further by enabling LLMs to use autonomous tools, which can call and dynamically adapt to external information. An example of this is the WebMD agent I developed that generates relevant WebMD search queries and scrapes the retrieved article content to create an evidence-based final conclusion. After gathering answer choices and explanations, I use LM as a judge automatic evaluation with Prometheus 2, an evaluation-focused language model with a strong correlation to human scores. Providing Prometheus 2 with expert written explanations from MedXQA and a customized medical rubric with a 1 to 5 scale, I collected quantitative assessment data, which is illustrated in Figure 5 at the bottom left of the poster. The models tested generally displayed similar accuracy of 82 to 85% and explanation quality of 4 to 4.3 out of 5, with these high scores supporting contemporary LM's viability as effective clinical assistants. Notably, MedGamma 3, a small medical domain-specific model, consistently underperformed in both metrics by a wide margin, but its results, despite its size, show potential for use in resource-limited settings. The agentic WebMD approach produced the highest average accuracy, but also the lowest average explanation quality. On the other hand, few-shot prompting produced the highest explanation quality with the competitive accuracy. These metric trade-offs for prompting approaches are illustrated by Figure 6a, and these specific prompting methods demonstrate how relevant reference information in prompts improves performance. A consistent trend in the data is that all methods consistently underperform in the speech-language pathology specialty, with none exceeding an accuracy of 37%, which points to a gap in existing LLM comprehension. I would also like to acknowledge the limitations of the study. First, I only use a single benchmark, which isn't representative of all clinical tasks and topics. Second, not having human annotators grade the LM responses can limit how accurate the evaluation process is. Could you go back to the slide?
Thank you so much, Rishi. Wonderful job. Now we will be moving on
I
to.
wasn't finished with my presentation. The slide just changed. I was asking for it to be moved back.
Can you say one more time, Rishi?
I wasn't finished with my presentation. The slide just got changed.
Oh, sorry about that. I'll move back. There you go.
Thank you. Future directions could also include expanding to more diverse tasks and benchmarks while also implementing unique prompting approaches and emerging LLMs. Additionally, future research could investigate specialty weaknesses, as seen in the study with speech-language pathology questions. Overall, we can conclude from these findings that there's a need to match LLM deployment to the specific needs of each clinical task and application, instead of just using general implementation methods. For example, when diagnostic accuracy is the highest priority, like in automated screening, for example, methods with higher correctness, such as the WebMD approach, can be advantageous. Additionally, clinicians can enhance model performance by utilizing relevant reference information when prompting for their tasks. These practices emphasize interpretability for safe, clear, and effective clinical workflow integration. Finally, I'd like to thank my mentor, Daniil, for his constant support throughout this research project. This project would not have been possible without him. And I'd also like to express my gratitude towards the OpenBio SRI team for this invaluable platform and experience. Thank you.
All right. Thank you, Rishi. Now we have Sharia who will be presenting.
Can you hear me?
Yes.
Okay. Hi, everyone. My name is Shoria, and I'm going to be talking about my project Plumpin, which is basically an attempt to make math, physics, and AI work together to figure out how air pollution moves around cities. So fine particulate matter is particulate matter with a diameter of 2.5 micrometers or less, or hence the name PM2.5. So those of you who took AP Environmental Science might know that these particles are smaller than the width of a human hair, but counterintuitively, they're more dangerous than any bigger particles because they can travel deeper into your lungs. And so PM2.5 actually causes over 7 million premature deaths every year, which is pretty much like wiping the population of a big city every single month. So how do people approach this problem? So traditionally, scientists predict air pollution using really detailed physics simulations, right? But these are, while they're really accurate, they're super slow, and therefore in a high-stakes scenario, it's kind of hard to predict it in time, and these can take weeks. On the other hand, data-only AI models are fast, but they don't always make physical sense. So what I mean by physical sense is, I'll explain it using a classic example. For those of you who take physics, this will be easier to understand. So like when you're modeling a spring's motion, right? An AI model might spit out like a nice looking output that seems fine at first, but then it might be physically impossible because it might not satisfy the damped harmonic motion equation, which the spring has to satisfy. But the AI model wouldn't realize this because it wouldn't have the physics embedded into it. So By combining the physics approach and the AI approach, you get a physics-informed neural network, which is what I built. And this model uses AI for speed, but it also listens to the laws of physics, so it's not making any implausible guesses. So for the city, I focused on Denver, Colorado, and collected 2 months of pollution and weather data. And I then split the city into a grid. And what I gave the model was the pollution sources, the wind conditions, and then a basic physics equation for how stuff moves in the air. And this is called the advection diffusion equation. Then I trained the model in 2 phases. So first, I just gave it the data without the physics. So this is kind of like warming up the model before it actually uses the physics equation. And this is a common practice in these physics-informed neural networks to make sure that the model also learns from the data and it doesn't try to only use the equations. And then the second step was embedding the physics equation into the model so it learns from the data while it's also making sure that all outputs satisfy the physics equation, getting the best of both worlds. So by the end of all this, my model had learned not just the patterns in the data, but also it learned a number called the turbulent diffusivity, which describes how fast pollution spreads in the air. And it found a value close to what real scientists get, which was 0.04. And you can see this in the figure with the black dotted line in the top. It kind of tapers off to that value. And then for the error, so how accurate was the model? So I got a mean absolute error of 2.35 micrograms per cubic meter and a root mean squared error of 2.91 micrograms per cubic meter. And both of these numbers are competitive with current literature while being computationally much more efficient. Like I ran this project on my 2020 MacBook in like less than 2 hours, contrasting with the vast amount of GPUs that other projects often take. So a few issues with my project. So first are with plumes. So plume, as you see in the name of the model, are like the times where there's a lot of pollution going on. So if you look in the bottom graph, you'll see a lot more underestimates than overestimates occurring in the predictions, which means that it's unable to predict the spread during high pollution events. And one thing I did to counter this is to upweight the penalty to 50 times the regular factor when it's looking at data that's high PM 2.5. And while it did help the model, it still wasn't perfect. And another downside with the physics-informed neural network is that the equation can't look at real world pollution spikes, such as wildlife smoke incursions or nighttime inversions, right? So because this is not covered, these extreme scenarios would not really be able to be tackled with this physics-informed neural network. So in general, just to summarize, my model predicted pollution levels in Denver with good accuracy, and it also learned the physics and a physically realistic spreading rate without me having to tell it what it should be. And this is, again, more realistic and more computationally efficient than purely data-driven models, which can help people breathe cleaner air. Thank you.
Thank you, Sharia. Now we will go on to Hannah.
Hello, everyone. My name is Hannah George, and today I'm going to be presenting on the emerging role of stimuli-responsive biomaterials in treating autoimmune diseases. Conditions like rheumatoid arthritis, multiple sclerosis, and inflammatory bowel disease affect millions worldwide and trap patients in a destructive cycle of chronic inflammation. Instead of resolving injury, the immune system mistakenly attacks healthy tissue, producing excessive reactive oxygen species and pro-inflammatory cytokines that damage cells, destroy tissue architecture, and perpetuate disease activity. This creates a self-reinforcing loop. ROS polarizes macrophages toward a pro-inflammatory M1 state. M1 macrophages generate more ROS and cytokines activate further immune responses, sustaining inflammation even after the original trigger is gone. Current treatments such as methotrexate and tumor necrosis factor alpha inhibitors can manage symptoms, but do so by broadly suppressing the immune system. This brings systemic toxicity, higher infection risk, inconsistent responses, and an inability to stop the underlying disease. Stimuli-responsive biomaterials take a fundamentally different approach. These are engineered materials that remain inactive under normal conditions, but activate when they encounter disease cues in inflamed tissue, such as high ROS levels, acidic pH, or elevated cytokines. By harnessing these signals, they deliver therapeutic action exactly where and when it is needed, sparing healthy tissue and minimizing systemic exposure. In fact, it is the deliberate pairing of smart material compositions with carefully engineered architectures that enables these systems to outperform traditional approaches, maximizing therapeutic precision while minimizing collateral effects. Two main strategies dominate this field. The first is ROS scavenging. Systems like cerium oxide nanoparticles, thiokidal-linked micelles, and phenylborate hydrogels cerium oxide nanoparticles can switch oxidation states to neutralize ROS continuously and shift macrophages toward the anti-inflammatory M2 phenotype promoting repair. In multiple sclerosis models, pegylated cerium oxide nanoparticles reduced oxidative stress and restored immune balance. Thioconyl-linked micelles release anti-inflammatory drugs only in high ROS environments, while phenylborate hydrogels degrade under oxidative stress to release antioxidants or drugs in a sustained way. The second strategy is cytokine sequestration, capturing pro-inflammatory cytokines like tumor necrosis factor alpha, interleukin 6, and interleukin 1 beta before they can drive further inflammation. Examples include biomimetic nanoparticles coated with immune cell membranes that decoy cytokines by presenting natural binding receptors. In rheumatoid arthritis models, neutrophil mimicking nanoparticles reduced inflammation and protected cartilage. Antibody functionalized nanowires can be injected locally to selectively expand regulatory T cells or activate immune cells as needed. Even more promising are multi-responsive platforms that integrate multiple triggers, such as ROS and acidic pH, to improve specificity and reduce off-target effects. Some even combine ROS scavenging with cytokine sequestration in one platform, addressing both major inflammatory drivers simultaneously. This AND-gate logic, where therapy activates only when multiple disease cues are present, could further reduce side effects and match the complexity of autoimmune inflammation. Clinically, this could mean patients receive localized therapies that act only during flares, lowering drug burden, reducing side effects, and preserving immune surveillance. Imagine a hydrogel that stays dormant in a joint until inflammation spikes, releasing therapy on demand, then returning to an inert state, a true step toward precision immunomodulation. Translating animal model results to humans, scaling up manufacturing, and ensuring long-term safety and biocompatibility remain as challenges, but with advances in diagnostics, personalized profiling, and built-in biosensors, future therapies could adapt in real time to each patient's disease activity. In the end, stimuli-responsive biomaterials are more than just a new drug delivery tool. They represent a paradigm shift in how we think about treating autoimmune diseases, replacing broad and blunt interventions with precise, adaptive, and patient-centered strategies. Thank you.
Thank you so much, Hannah. Now we have Daniel who will be presenting.
Hello, can you hear me?
Yes, you're good.
Okay. Hello, my name is Daniel Rue, and I'm excited to share my research on the behavioral health impacts of drug policy on young people in the United States. And this project was conducted as part of the Student Research Institute in collaboration with Jacqueline Chess from UC Berkeley. Over the past 2 decades, US drug policy has been moving away from purely punitive approaches toward more public health-oriented models, such as decriminalization and legalization. Traditional punitive policies often led to high incarceration rates, long-term stigma, and limited access to treatment without necessarily reducing drug dependency. Decriminalization, on the other hand, generally removes criminal penalties for possessing small amounts of drugs for personal use. While the most common example is marijuana reform, the actual behavioral health impacts, especially for young people aged 12 to 15, are still not well understood. This review aimed to synthesize existing research and identify how policy design can either improve or harm youth outcomes. I conducted a systematic literature review using PubMed, focusing on peer-reviewed studies in the US, with Portugal's public health-based model as a comparison point. My search terms covered drugs, behavioral health, youth demographics, location, and policy language. Now, out of 262 articles, I excluded those without a clear policy framework, youth focus, or behavioral health outcome. After full text screening, 16 high-quality studies were included in the final review. For more detail, we can see the Prisma chart that details the screening process for the sources and how I eliminated most of the sources from the beginning. The results fell into 5 major themes. The first was youth initiation and substance use trends. About 80% of drug users start during adolescence, and 11% meet the criteria for a substance use disorder before age 18. Now, recreational marijuana legalization was linked to increased use among certain groups, especially females and college students, while medical marijuana laws showed mixed effects, sometimes even reducing use among younger teens like 8th graders. Second, behavioral health impacts. Frequent cannabis use during adolescence was tied to lower academic performance, underemployment,
cognitive deficits, depression, anxiety, psychosis, and a higher suicide risk, especially when starting before the age of 16. Roughly 17% of adolescent cannabis users developed cannabis use disorder, often alongside ADHD or depression. Third, for policy structure, Portugal's decriminalization, which was paired with strong harm reduction and treatment systems, kept youth use stable, boosted treatment uptake, and lowered overdose deaths. The US, by contrast, which often follows a commercialization model with aggressive marketing, high potency products, and weak regulations, factors linked to greater youth use and lower perceived risk. Fourth, treatment access. Only 10 to 15% of adolescents in the US who need substance abuse treatment receive it. Portugal's integrated approach reduced stigma, and improved continuity of care, highlighting that policy can directly influence whether youth get help. Disparities. Black youth face higher arrest rates for marijuana possession despite similar usage rates to white youth. Rural, Indigenous, and LGBTQ plus youth often experience higher risks and worse access to care. And these are gaps that can widen after legislation without targeted interventions. Now, these findings point to a clear conclusion. Policy design is just as important as the policy type. Decriminalization alone is not some magic fix. Without strong public health safeguards, commercialization can worsen behavioral health risks, especially for vulnerable youth. Portugal's model shows that when decriminalization is paired with prevention programs, integrated treatment, and sustained public education, it can improve outcomes without driving up youth use. In the US, however, fragmented healthcare systems, state-by-state regulations, and market-driven legislation have made it harder to achieve those same results. And social context also matters. Rural communities often have fewer treatment resources, meaning increased availability of high-potency cannabis can hit them harder. Communities of color, already disproportionately policed, may benefit from fewer arrests under decriminalization, but they still face structural barriers to care. Finally, the research gap is significant. Most studies focus on cannabis, leaving little evidence on the effects of decriminalizing other substances like psychedelics or opioid alternatives. This is especially relevant now as states like Oregon and Colorado are experimenting with regulated psilocybin services, and harm reduction strategies for opioids are expanding nationwide. In summary, cannabis decriminalization can reduce criminalization and stigma while improving treatment access, but only when prepared with prevention, equity-focused interventions, and robust regulation. Future research should expand beyond cannabis, examine long-term mental health effects, and explore how policy design can protect marginalized populations. By learning from models like Portugal's and avoiding the pitfalls of unchecked commercialization, policymakers can better align drug policy with public health goals. Thank you for listening, and I look forward to discussing how evidence-based policy can create healthier futures for young people.
Thank you so much, Daniel. Now we have Aishini who will be presenting. Aishni, are you here? All right, it doesn't seem like she's with us, so we can skip over, and then if she joins at the end, we can come back to her. All right, so now we have Thomas.
Hi, thank you. Hi, so I'm Thomas, and I'll be presenting a computational study done with my mentor Ethan Neal from the University of California, Santa Cruz. So we studied the use of a feed-forward neural network on the knapsack problem and compare it to the standard dynamic programming solving method. So first, I'll explain the problem and why it's really interesting to study. So it's an NP-complete problem, meaning there's no optimal solution that runs in polynomial time, even though the solution is verifiable very quickly. For example, Sudoku is also NP-complete. It's very hard and slow to solve, but once you have the solution, you can verify it very quickly. So the problem consists of choosing the best items to add to a knapsack or a backpack. You have a set of items with different weights and values that can be picked to be added to a knapsack that has a maximum weight capacity. The goal is to maximize the total value of items inside the knapsack while staying under the weight limit. So this problem is normally solved using an algorithm that uses dynamic programming. It always solves the problem optimally, but takes very long to solve. If you just ask it to solve the problem when there are 1,000 items to choose from and the maximum weight capacity is 10,000, it will take about 4 days and 6 hours with a modern computer. So this is the reason we experimented with another method to solve this problem. in a much quicker way, hopefully, using a neural network. So another motivation for this is the lack of research investigating the impact of noise in the problem instances, specifically in the weights of items. So as a result, in this study, we compared the standard dynamic programming method to custom-built feed-forward neural network on three metrics: accuracy, time complexity, and robustness to noise. So I'm now going to explain our methods. So first, we generate a large number of solved knapsack problem instances with dynamic programming. This data is then fed to the neural network for training. And finally, we test the neural network on a set of new problem instances and compare the results and the solution to the optimal solution from dynamic programming to measure our accuracy, which is basically if the same items are selected and if the total knapsack solution is accurate. So this whole methodology is shown in figure 2. And the process is similar when studying robustness to noise. So we first add Gaussian noise in the item weights. These noisy problem instances are solved by dynamic programming and then fed to the neural network to train with. So we train it with noisy problem instances. Then we test both the neural network and dynamic programming on new noisy problem instances. And both of these solutions are used on the true problem instance without noise, and their optimality ratio is computed. So basically, our goal is to compute how far off both of the method solutions are from the original problem instance solution. And the results were extremely impressive. The neural network solutions' accuracies were constantly above 91% and reached a maximum of 96%, while the neural network time complexity is only polynomial, specifically n times log of n compared to the n times 2 to the k complexity of dynamic programming, where k is the number of bits needed to represent the maximum capacity. So this means the dynamic programming solution is exponentially slower than using a neural network, as shown in figure 3. So the neural network also performed much better on robustness to noise than dynamic programming, as shown in figure 4. The neural network's optimality ratio is on average 1.73 times higher than dynamic programming over different Gaussian standard deviations, meaning it is much less sensitive to Gaussian noise in the weights. Unfortunately, due to limited computational power, we could not further investigate the convergence of the neural network's loss and accuracy, and maybe we could have reached higher loss. Nonetheless, we also present a mathematical method to estimate accuracies and loss depending on the number of samples the neural network is trained with. And similarly, we only used a smaller neural network of 2 layers with 640 total neurons. However, a wider and deeper neural network trained on more number of samples might have performed better in accuracy, but we simply could not do it because of computational constraints. So as a result, it is clear the neural network can definitely replace the standard dynamic programming method in many contexts, especially real-world applications where the dataset is usually very noisy. And by the way, that problem instance would have taken the dynamic programming method, over 4 days to solve. The neural network would have solved it in about 10 seconds. I would like to thank my mentor, Ethan Mule, for all his help and guidance throughout this program. This research could not have been possible without him. And also thank Harvard's Open Bio Student Research Institute that provides the amazing opportunity to conduct this research. Thank you.
That was a great job, Thomas. Thank you so much. Now we will have Driti, who will be presenting.
Imagine your brain as a busy city. Every night, garbage trucks move through the street, clearing away waste so everything is running smoothly by morning. Now, picture what would happen if the garbage trucks stopped working. Trash starts piling up, roads are blocked, and soon the whole city is losing efficiency. This is similar to what happens inside the brain of someone with Alzheimer's disease. When the brain's waste clearance system, the glymphatic system, and its cleanup crew, the microglia, break down, harmful waste begins to build up. Hi, I'm Diti Kintipudi, and I'm from Northville High School in Northville, Michigan, and this summer I, alongside my mentor, Hannah Yoder, conducted a systematic literature review on the interplay between microglia and the glymphatic system in Alzheimer's disease, also known as AD. AD is a progressive brain disorder that gradually destroys memory and thinking skills, and eventually the ability to carry out even the simplest tasks. In fact, the World Health Organization reports that AD accounts for 60 to 70% of Alzheimer's of dementia cases, imposing heavy societal and healthcare burdens on patients and their families. AD is marked by amyloid B, AB plaques, tau tangles, and progressive cognitive decline. For decades, most treatments have tried to reduce the production of these proteins, but with limited success. That's when I learned about the extensive research on the role of the glymphatic system and microglia in waste clearance. Therefore, understanding how the body clears accumulating proteins is crucial for developing effective interventions. Therefore, I conducted this systematic literature review to contribute to insights for developing therapies to increase toxic clearance efficiency in human brains. So using the PubMed and Google Scholars database, I identified several articles of interest. And out of the 1,422 articles I initially identified, I ultimately chose 51 articles for my review, and here are some of my main takeaways. Microglia are the brain's primary immune cells, constantly patrolling their surroundings to respond to pathogens and damage. In AD, however, they shift from this protective state into a disease-associated state, marked by chronic inflammation, reduced ability to clear AB and tau, and even damage to healthy synapses. Instead of safeguarding the brain, they end up sustaining a toxic environment that accelerates neurodegeneration. As shown in Table 1 on my poster, these microglial changes are closely tied to glymphatic dysfunction. In AD, AQP4 channels on the astrocyte end feet lose their organized placement, disrupting the directional flow of cerebrospinal fluid. Perivascular structures weaken, limiting waste clearance, and communication between astrocytes and microglia break down, making both systems less effective. The glymphatic system itself is a brain-wide clearance pathway that moves cerebrospinal fluid along arteries, through brain tissue, and out along veins to remove waste. This process relies on AQP4 channels positioned at the ends of astrocytes around blood vessels. In a healthy brain, as depicted in Figure 1, this arrangement supports efficient fluid exchange and rapid waste removal. But in AD, the misplacement of AQP4 channels slows fluid flow down and waste clearance, often before large amyloid plaques even form. Figure 2 illustrates the vicious cycle linking these systems. Impaired glymphatic clearance allows toxic proteins to accumulate, which activates microglia. The resulting inflammation further disrupts astrocytic function, worsening lymphatic flow, and fueling even more protein buildup. Lastly, as shown in Figure 3, the literature highlights 4 main therapeutic approaches: restoring communication between astrocytes and microglia, breaking the cycle of inflammation and impaired clearance, improving fluid dynamics by targeting sleep circadian rhythms and vascular health, and most importantly, dual targeting, simultaneously modulating immune activity while restoring glymphatic function. The Venn diagram highlights where these strategies overlap, showing that dual targeting appears most effective early in disease progression when the systems still have the capacity to recover. Acting early could help restore both waste clearance and immune balance before irreversible damage sets in. Overall, microglia and the glymphatic system work in a two-way relationship, each affecting the other in Alzheimer's disease. This interaction drives both inflammation and protein buildup. The evidence suggests that targeting both systems together could be more effective than focusing on one alone. Most of what we know comes from mouse models, so the next step is developing better tools to study these dynamics in the human brain. Finally, I would like to thank my mentor, Hannah Yoder from ICANN School of Medicine at Mount Sinai, and the SRI for giving me the amazing opportunity to conduct my research. Thank you.
Kirti, that was great. Thank you so much. And now we have our final presenter, Arjun. So whenever you're ready.
Right now, more than 10 million people worldwide are living with Parkinson's disease, and for most of them, diagnosis came years too late. Why? Because the disease hides in plain sight. Subtle changes in walking patterns or speech can appear up to a decade before the tremors and stiffness that's associated with Parkinson's disease today. But these early signs are so faint, they're almost impossible for a human to notice. Hello, my name is Arjun Subramanian, and today I'll be presenting my project, Unified Multimodal Deep Learning Framework for Preclinical Detection of Neurodegenerative Diseases via Spatiotemporal Gate Dynamic and Acoustic Biomarkers. It's a long name, but the idea is simple. Use deep learning to uncover these hidden patterns before the damage is irreversible. These diseases don't happen overnight. They actually progress quietly and slowly. In early stages, a Parkinson's patient might just only take slightly uneven steps or pronounce certain vowels differently without realizing it. But the opportunity here is huge. These are non-invasive biomarkers. No blood tests, no MRI machines, just sensors or microphones. So it's possible to do this from the comfort of your own home.
And if we detect them early on, we can start treatment years sooner, slow down the progression, and improve quality of life. The challenge is that these changes are subtle and faint, which is exactly where deep learning excels. My idea was to fuse two complementary streams of information, gait force signals, which reflects motor control, and speech patterns, which captures both motor and cognitive changes. For the gait model, I used a CNN plus transformer to model complex spatiotemporal patterns. For speech, I extracted acoustic features like MFCCs and fed them into a multi-layer perceptron. By pairing each modality with the architecture it's best suited for, we boosted accuracy and made the system far more generalizable. I worked with two specialized datasets, GATE-NDD from PhysioNet, which had raw foot sensor data from Parkinson's, ALS, Huntington's, and healthy controls, capturing stride intervals, swing times, and asymmetries. And I also used the Italian Parkinson's speech dataset, which were recordings of vowels, syllables, and phrases from Parkinson's patients and matched controls, allowing for the extraction of MFCCs, jitter, and shimmer data. For pre-processing, the speech was transformed into Mel spectrograms and MFCCs, while the gait signals were converted into force curves, showing the pressure patterns over time. This gave the model a lot of time series inputs for both the movement side and audio side of things. Well, here's how it works though. Both modalities start with a CNN feature extractor to identify spatiotemporal patterns. From there, the features enter a transformer encoder, which uses positional encoding to preserve the sequence order, applies the multi-head attention to connect patterns over time. For example, linking stride variability with subtle vowel distortions, and it also uses feed-forward layers and layer normalization for stability and scalability. Finally, the model outputs a probability score through the classification head, trained using binary cross-entropy loss to match predictions with ground truth labels. When the model was trained on gait alone, the model reached about 94% validation accuracy, steadily improving over the 50,000 steps. When it was trained on speech alone, it reached over 92% with smooth learning curves and minimal overfitting. When I fused both models, though, accuracy improved further and generalization got stronger, proving that gait and speech capture complementary insights that others can't fully provide on their own. And when it comes to future work, I believe the next steps are to integrate more modalities such as facial expression analysis, heart rate, or EEG, to explore more fusion techniques such as late fusion or hybrid fusion, and to also expand to more multilingual speech data sets for broader language coverage and further accessibility for other third world countries. And I also want to compress the model for mobile deployment for once again better accessibility and for more people to be have access to it. So in summary, my project shows that tailored modality-aware AI systems can detect the early signs of disease with high accuracy, long before a diagnosis is possible today. That means earlier intervention, better outcomes, and a scalable, more affordable solution for both clinics and homes. This is not just about predicting diseases. It's about giving people more healthy years to live their lives. Thank you so much for your time.
Thank you so much, Arjun. That was a great way to wrap up all of our presentations. Thank you guys so much. You guys really did a really impressive job, and all of your projects, your lit reviews have been wonderful, and it's been really great to hear about all the work you've been doing this summer. So it seems like we're about 20 minutes ahead of schedule, which is great. The other rooms are also getting pretty much ahead of schedule as well. So you guys are free to join the main session and wait until they start the final closing remarks, and then from there you guys are free to go. So you guys can leave the room.
Oh.
Thank you guys so much.